Welcome to the SAN Science Experience, your source for the most current and impactful research and preprints in science. Please like this video and subscribe. It really helps support the SAN Science Experience. Today, we're diving into a really fascinating study. It uses these cutting-edge tools, like AlphaFold 2.
The AI structure predictor, yeah.
Exactly, to explore how metabolism, something so basic, has actually shaped enzyme structures. And get this, over 400 million years.
It's quite the timeline.
Yeah.
And this work really paints a kind of hierarchical picture. It shows enzyme evolution isn't just random. It's driven by their function, sure, but also deeply influenced by the organism's whole metabolic niche, its lifestyle, you could say.
Yeah.
And, the network architecture, how much it costs to build the proteins, even specific molecular interactions, it's all connected.
It sounds incredibly complex. So this grand blueprint, how did they even start to piece it together? What was the scale here?
Well, the scale was, frankly, huge.
Yeah.
They analyzed, 11,269 enzyme structures.
Wow.
Yeah. Some predicted by AlphaFold 2, others known experimentally.
Mm-hmm.
And these covered 361 reactions in 225 pathways.
Okay, so a massive amount of data points.
And not just in one bug. They looked across 26 different yeast species, all within the Saccharomycotina group, spanning that huge evolutionary time.
Right, 400 million years.
Yeah.
So what's the key insight from connecting all that structural info with metabolism?
Well, what's really neat is how they managed to link changes in the enzyme sequences, the divergence specifically in regions that kept their overall 3D shape.
Mm-hmm.
They linked that to different metabolic properties. So it's this blend of structural biology and evolutionary genomics. And that let them sort of tease out the rules, the constraints acting at all these different levels, species, pathway, molecule.
Okay, let's break that down. So starting at the species level, does what a yeast eats actually change its enzymes over time?
It absolutely does.
Ugh.
The study showed pretty clearly that metabolic specialization, like does the species mainly ferment glucose or can it use something like xylose? That directly shapes how its enzyme structures evolve. So for instance, enzymes from yeasts that are really good glucose fermenters, they actually showed smaller differences in structure compared to the standard lab yeast, cerevisiae.
Less change, okay. Like they're highly optimized.
Kind of like that, yeah. Finely tuned. But then you look at enzymes for, say, using xylose.
Yeah.
Their conservation, how much they stayed the same, varied a lot, depending on whether the species could actually use xylose.
Got it. So lifestyle directly impacts enzyme stability. What about the pathway? Does the enzyme's neighborhood matter too?
Oh, definitely. Enzymes in those really central pathways, think making DNA bases, essential amino acids, or core energy stuff like glycolysis, the TCA cycle.
The big ones.
Right, the big ones. Those tend to be super conserved. Their structures barely change.
Yeah.
But then you look at pathways like lipid metabolism or adding sugars to proteins, glycosylation. Those enzymes were much more diverse. They changed more.
So why the difference? Why are some pathways so much more rigid, evolutionarily speaking?
That's a great question. And the study suggests it's not just about how much stuff flows through the pathway. It might be more about the variability of that flow.
Variability? How so?
Like how consistent the demand is on that enzyme or pathway. And maybe enzyme processivity, how smoothly it works. If the workload is really unpredictable, maybe the machinery needs to be more robust, more unchanging.
Ah, okay, that's subtle. It's the consistency of the job, not just the volume. Okay, now this is where I think it gets super interesting down at the molecular level. What's happening with the actual enzyme atoms?
Right, so down there, you see this clear hierarchy. It's about cost optimization versus conservation. Enzymes that are rare, low abundance, maybe only needed sometimes, they tend to be more diverse structurally and also more costly.
Costly
meaning they use amino acids that take more energy for the cell to make. And this cost saving, this optimization, happens mostly on the enzyme surface.
Oh, wait, the outside evolves faster.
Exactly. But the core of the enzyme, much more conserved. And here's the kicker, the really critical bits, the binding sites.
Where the action happens.
Precisely. Where the substrate binds, or maybe regulatory molecules, those allosteric activators, those spots are the most conserved. And interestingly, they're the least optimized for cost.
So evolution doesn't skimp on the important parts, even if they're expensive to make. Those binding sites are just locked in.
That's exactly what it looks like.
Yeah.
They found these highly conserved amino acids tend to cluster together, and yep, those clusters are usually these crucial binding or interaction sites. Think pyruvate kinase. Its key sites are incredibly stable across all those species all that time. The surfaces can change, adapt, optimize cost, but those functional hotspots, rock solid.
Wow.
It's like evolution is this really smart engineer, cut costs where you can on the surface, but never ever compromise the critical functional parts.
So yeah, this deep dive really drives home how metabolism isn't just, background chemistry. It actively constrains and guides how life's essential machines, these enzymes, evolve. Function dictates form, right down to the atoms, over mind-boggling timescales. It's pretty profound when you think about it that way. What really stands out to you from all this? I mean, the potential applications here seem huge, right? For understanding enzymes better, maybe even engineering them?
Oh, definitely. Understanding these evolutionary rules could be massive for annotating enzyme function, maybe predicting it better from sequence or structure alone. And yeah, from metabolic engineering, designing new pathways.
Yeah.
This gives us some design principles from nature itself.
Absolutely fascinating stuff. Well, if you found this deep dive as mind-bending as we did, please do remember to like this video and subscribe to the Science Experience for more cutting-edge science insights.